Okay, thanks everybody. Lovely to see so many people here turn out for our session. I'm Sanjay Shukla. I'm the CEO of aTyr Pharma. I'm thrilled today to introduce Dr. Dan Culver, who's going to talk a little bit about our therapy, Efzofitimod, a potentially emerging treatment for sarcoidosis.
Thanks, Sanjay. Thank you, Sanjay. Can everybody hear me okay? Perfect. It's wonderful to see so many people here. Thank you for all making time. I know these meetings are busy, so I'm very happy to be able to talk to you about something that I'm excited about, and it's nice to see so much excitement is shared here in the room. I'm going to talk about Efzofitimod a bit today, and as you all know, there was a phase 1b/2a trial recently published on this. And this is a little bit of a backwards story because this started off with a trial and now the science is filling in. And so I'm going to talk a little bit about the science that's being learned by aTyr behind this. But I want to caveat this with a couple of things. Number one, the science is still being learned. It's still being discovered. I'm going to just give you a little bit of what's been found so far about what this molecule and what this target could be doing in sarcoidosis. Second thing about this is that I'm not an immunologist. I know enough about how to pronounce some cytokine names, but if there's any hard questions, we'll refer it to Dr. Prase, wherever she is around here, or any of the other scientists that are here. Okay, well, so without further ado, when I was in medical school, I learned that tRNA synthase is really constructed proteins, and I thought that was the whole job that they had. But there's a whole area of biology I was unaware about, discovered by a guy called Paul Schimmel, which is actually the genesis for this company. And this area has to do with splice variants of tRNA synthases being secreted and having extracellular non-canonical functions. And those functions include downregulating of immune responses. And so there are a number of both genetic and autoimmune diseases where there's dysregulation of this mechanism, and those result in inflammatory diseases. So this particular Efzofitimod actually is a smaller version of a histidyl in our tRNA synthase, not the full-length version, but this is utilizing the anti-inflammatory properties that we think exist in this class of tRNA biology. So to refresh your memory, there was a trial using this particular medication. It was called aTyr 1923 at the time. And here's the consort plot from that. And this is published in Chest if you're interested in looking at it. As you can see, there's a couple of things you'll notice. Number one, this is a very small trial. This is a multiple ascending dose trial. And I think actually aTyr deserves a good bit of congratulations for pulling this off during COVID. You can see the dropout rate was not insignificant, but most of the dropouts were due to study sites having trouble operating clinical trials during COVID or patients not wanting to come back in. And so in fact, to get this trial across the finish line during COVID, I think was a pretty major accomplishment. In fact, the reviewers didn't like the size of this trial, but nonetheless, I'm going to show you this is a positive trial, surprisingly enough, with this small sample size. Now, this trial primarily was designed to assess safety of the medication. I can tell you, without showing you the data here, that there is no safety signal, either total AEs or SAEs, for any of these study groups compared to placebo. Of course, again, small numbers, and we'll see in the larger registrational trial whether that pans out. But so far, this looks to be very well tolerated. I want to also point out that one of the concerns about this has been the induction of antidrug antibodies. And could you induce autoimmunity like anti-Jo1 antibodies? And would that induce autoimmunity? I can tell you that none of that was seen in this particular trial. I would also say that this is being monitored very closely, of course, by the sponsor, but the FDA is also quite interested in it, and that'll be something that we'll have to keep an eye on. I personally don't look at this as much different than any other protein we give to people. It could always induce anti-protein antibodies. That can always be a side effect of any kind of therapy like this, and so that's something that we have to screen for and monitor. So what about the outcomes of the trial? Here's the steroid reduction. And I put this up here first because I want to point out that this is the emerging paradigm for sarcoidosis clinical trials. We used to be very much aligned with IPF, and we used to just focus on the vital capacity. But in point of fact, that was not the right model, not the right paradigm for thinking about sarcoidosis clinical trials. And what happened in this trial really was a reduction of steroids during the course of the trial, and then some of the key endpoints included the ability to reduce steroids. And you can see by various different ways of measuring steroid reduction there that there's a dose response curve, and that's consistent across the doses. And by different ways of measuring steroid reduction, we're able to demonstrate that dose response curve. There were 2 patients, actually 3 patients, that were able to get off of prednisone altogether. That occurred at the investigator's discretion. I can tell you that one of those was in the placebo group, and that person, during the trial, got worse again and had to go back on steroids. But I think that this is not a super huge signal, but it certainly is tending in the right direction of what we would like to do with our patients, given the known toxicities we have with long-term management with corticosteroids. What about lung function? Here's the forced vital capacity data. You can see again a dose response. This is not statistically significant. However, I would say that with the effect size, this is not too dissimilar from what we saw in the original infliximab trial that was published in the Blue Journal more than 20 years ago. And so these are the sorts of effect sizes we might expect to see in a chronic sarcoidosis population who's on some background therapy. And remember that this effect is happening during the course of steroid reduction. If you think that steroids might improve FVC, in fact, you're swimming upstream and trying to demonstrate this effect. And I think that the best actual clinical endpoint, or at least the most robust one in this, is really the PROs. And here are several different PROs. You can see them listed on the left. There were a variety of other PROs tested here. You can see that a number of these, those are the bolded ones, achieve statistical significance, and that many of them, which are the ones shaded in green, actually achieve differences that are greater than the minimally clinical important difference. And so again, you see a dose effect curve. You see responses that are important to patients. And remember that this is in the context of steroid reduction. And so being able to demonstrate that at the time you're reducing steroids either means that the medication is really causing some benefit or that the steroids were causing some negative effects on some of these quality of life markers. And I certainly think that some of that could be part of the story here. So we have a medication now that looks to be pretty safe and well tolerated in a small number. We have across a number of endpoints, FVC, PROs, steroid reduction, a dose response curve, and we also have some of those effects that are beyond what patients would perceive, beyond the minimal important clinical difference. So I think that in a sample size of this magnitude, this either means that, and I've told the aTyr folks this, they're either really lucky or this is a really good medication. So there's currently a registrational trial going on right now. I know that several of you are PIs and are involved in that. And I want to point out the importance of this trial. And it's not just because I like this particular molecule, but I want to point out that this is important to the community who care for sarcoidosis patients. We're at a place now where there are several industries, several companies who are getting interested in sarcoidosis. However, there's no approved medication for sarcoidosis in the modern era, and so there's no clear-cut pathway for regulatory approval for sarcoidosis medications. If we get a first medication, it could be this one, it could be a different one, across the finish line in a way that demonstrates the correct regulatory pathway, the same way we saw for IPF, the same way we saw for pulmonary hypertension, that will lead to more interest in the field. It'll lead to the better development of clinical endpoints, more understanding about the endpoints, and importantly, more understanding about the biology. So I think that's one of the reasons why this is so exciting and why I think we as a group, as a community, need to really push to make these trials go forward. So I'd said before that I think that steroid withdrawal is the key new paradigm for how we manage sarcoidosis trials. I'm going to put these glasses on and see if this helps. Wow, that doesn't help at all. I'm going to come over here. So I think one of the things that we didn't recognize before is that sarcoidosis patients who are managed with chronic disease therapy tend to stay stable. It's not like IPF. And so to demonstrate biologic effect of a medication, we really need to withdraw that background therapy. The problem with sarcoidosis and what makes it really hard to design a trial is that it takes a while for people to do poorly. And the estimates are somewhere between 20 and 80% of people will relapse after withdrawing steroids, and that about 50% of those relapses will happen within the first 6 months after withdrawing steroids. But if you imagine drawing a Kaplan-Meier plot of the time to relapse after stopping steroids, and then you imagine the length of a trial, and you imagine the effect size you're trying to get, and you're thinking about powering a study, you right away start to imagine that there's a significant difficulty for conducting trials in sarcoidosis. Now, one thing that people say is it really ethical to withdraw steroids from patients with sarcoidosis? How can that be ethical? And I've had people in my own institution ask me that. And I always ask them one question: have you asked the patients? Because if you talk to patients, it's unanimous. They feel very strongly that anything that might allow them to withdraw steroids, even if it's part of a trial and they're on placebo, is acceptable. There's not a patient that I know that's happy about being on steroids. So just to hammer a point home, here's some data that show current state of randomized controlled trials and any clinical trial in sarcoidosis and several other different diseases. And I'll point out, of course, that some of these are more prevalent than sarcoidosis, but some of them are not. IPF is no more prevalent than sarcoidosis. I would submit to you that pulmonary hypertension is no more prevalent than sarcoidosis, unless you start taking really mild cases and looking in other ways. And I just put in yellow here the number in PubMed of randomized controlled trials. And I think the point is, number one, sarcoidosis has by far the fewest of these. That relates to endpoint, it relates to us not enrolling, and it relates to not good understanding of the biology. But I also want to point out that sarcoidosis tends to have much fewer randomized controlled trials. And as a community, we've accepted a lot of data that is not randomized controlled, and we've based our decision making on that, and we have not insisted to sponsors, to FDA, to other sources that we need better data for managing our patients. And so that's a plea again to support randomized controlled trials. That's the only way we're really going to move forward in this field. So now let me pivot a little bit to Efzofitimod and what we think it might be doing. Here's a cartoon. This is sarcoidosis. And as you all know, sarcoidosis is a cell-mediated immune response. There's some triggering antigen. I tend to believe there's probably more than one triggering antigen. I tend to really think that maybe the antigen's not the important part of the story. And we've always thought about sarcoidosis as being primarily a cell-mediated response where the lymphocyte is the lieutenant, the colonel, the general, the most important piece of it. And I think that's what's being recognized more and more now, is that this is both an adaptive response and still importantly an innate response. And that the innate responses in this type of chronic inflammation are very, very important. And I would point out to you that if you look at, for example, BAL in patients with chronic sarcoidosis, not with Lofgren syndrome, quite often you find a very low lymphocyte count. And that doesn't mean lymphocytes aren't important. You have to have lymphocytes in order to drive this inflammation and to perpetuate the inflammation. But I want to say that that's not the only part of the story, and that there are probably multiple ways to tackle granulomatous inflammation. I'm going to show you this thing from Dr. Krauser. This is lymph nodes from sarcoidosis patients, and this is the dominant gene network in sarcoidosis lymph nodes compared to control. And this is very statistically significantly high overexpressed in sarcoidosis patients. And the key, one of the key nodes in the middle of that is STAT1. STAT1 is the canonical signaling target of interferon gamma signaling. This is a highly Th1 biased interferon gamma signature disease. On the bottom is immunohistochemistry looking at phospho-STAT1. And so we can start to use these things to untangle the biology of sarcoidosis. On the right is evidence from single-cell-seq in skin biopsies of patients treated with a JAK inhibitor, tofacitinib, for cutaneous disease. And you can see a number of JAK-dependent and JAK-independent signals that are modulated in sarcoidosis and that are specifically important in the pathobiology of sarcoidosis. And I just want to point out again that this includes both signals that are primarily related to the adaptive immune response, as well as signals that are seen very much in the innate immune response. So again, innate immune responses are important. So how does that relate to Efzofitimod? So efzofitimod actually is a ligand for a receptor that I never heard of before this called neuropilin-2. Neuropilin-2 turns out dimerizes with a number of other receptors like VEGF receptor and semaphorin receptor, and it's in this complex you can see over here on the right. And in fact, neuropilin-2, as I said before, is expressed on a number of cells in the body and has effects on dampening immune responses. It does look like efzofitimod binds primarily to the beta-1 subunit, and as such, it doesn't seem to interfere with VEGF signaling. It doesn't seem to interfere with semaphorin receptor signaling, and I'll show you a little bit of data about that. Here's just evidence showing that it's the binding is highly selective. Here's a control here in the black showing very poor binding as compared to efzofitimod that's in the green. So this is selectively binding the neuropilin receptor. There's a screen of over 4,000 different other proteins. There doesn't appear to be binding of any other proteins, cell surface receptors in that screen. So this looks like it's highly selective for neuropilin-2. What about sarcoidosis? Here are some stains from sarcoidosis patients. In the middle is immunohistochemistry. If you're not used to looking at immunohistochemistry, you just look for the brown. The brown parts are in the middle of the granulomas. That's where the neuropilin-2 receptor is found. And in fact, if you do in situ hybridization and look a little bit more closely, you can see on the right that these are primarily concentrated in the epithelioid cells, which are macrophage-derived cells, and in the multinucleate giant cells. So we think this is primarily a myeloid cell receptor. That's where it's mostly found, at least in sarcoidosis patients. I'm going to have to stand really close to this screen. I apologize. So neuropilin, where is it in the body? Where is it in the sarcoidosis patients? One thing that we see is that as monocytes differentiate into macrophages, either into M1 type macrophages, that's on the top, you can see the flow where there's a shift to more neuropilin receptor with increasing differentiation, or dendritic cells even, you can see increasing neuropilin receptor expression. If you look at sarcoidosis patients, and some of this data was presented yesterday, I know, it is overexpressed in sarcoidosis patients in general compared to healthy controls. And if you look at those monocytes, those are doing the same things that we think are important in sarcoidosis. They're overproducing CCL2, they're overproducing TNF, they're overproducing IL-6. And in fact, when we looked at the sera from the clinical trial, we see again a dose response on those particular cytokines, and there are a number of other ones that were looked at, but these are just demonstrative, showing that these things are indeed reduced by treatment by efzofitimod in a clinical scenario. So we know now that NRP2 is found in granulomas. We know that as monocytes differentiate, it's expressed on monocytes, and that those same pro-inflammatory monocytes produce these cytokines, and that we saw reductions of those in our clinical trial. So how is it binding to neuropilin? What's it doing? It does appear, as I said before, that it binds to selectively to neuropilin, primarily the beta-1, but it really is binding into this tertiary structure, this propeller structure, where alpha-2, beta-1, and beta-2 domains come together. And you can see here the specificity of that on this bottom plot. If you look at other ligands like VEGF or semaphorins, you don't see binding, and on the right side plots, I just want to point out that as you add more efzofitimod, you don't see these curves really shifting. And so that's showing by luminescence assay that as we add in increasing concentrations of efzofitimod, we don't change the ability of VEGF to signal, we don't change the ability of semaphorins to signal, and so we don't think that there's likely to be pathway interference with those other pathways. As monocytes differentiate, we see that neuropilin-2 is expressed more. I already showed you those data. And we see that the monocytes become more spread. They get more of a macrophage phenotype, and that they achieve an M1 polarization. And I know that M1 and M2 are, number one, difficult to understand in human disease, and number two, a bit controversial in sarcoidosis. And there have been some groups that have published M2 bias and some groups that have published M1. And I think that probably there's a lot of plasticity, and it's probably context dependent, it's probably temporal dependent. If you do pathway analysis by single-cell-seq and look at what's happening on these cells, you can see a number of pathways that are expressed as you differentiate in these human donors, and you see increasing NRP2 expression. And those are pathways, several of those that we would know are important in sarcoidosis. If you look at inflammatory gene expression and cell surface receptors, again, in those differentiated monocytes, you can see, again, CCL2 and TNF-alpha are responding to efzofitimod. You can see that the cell differentiation is different. So CD14 is lower. You can see that there's more of a right shift in the untreated ones than the treated ones in the middle bar. And then you can see that with efzofitimod, you can see that that's reduced. And so, again, we think that this is somehow changing the differentiation of monocytes into those pro-inflammatory macrophages, and then maybe also interacting with actually what's happening in the granuloma. So putting all that together, what are the targets that we know about so far, and what are the targets that we don't know about? And here's a graph, and I won't go through all of those, but if you look at monocyte-derived macrophages and look at what's seen, as well as you look at the clinical trial data and you look at both cytokines and receptors, you can see that several things that we think of as important in sarcoidosis biology seem to have a signal. And so these will be areas for future investigation. This is by no means the end of the story about understanding the relationship between neuropilin, inflammation, and then how efzofitimod might influence that particular thing. I think I have one more slide about this. This is just looking at immune recruitment. This is using a whole different model. This is not a sarcoidosis model. This is just a lung injury model with LPS. And just to show you that this probably has more broad generic effects. So here's an LPS model looking at recruitment to the lung of macrophages, lymphocytes, and neutrophils, and you can see that there's a dose response for efzofitimod for each of those things. And so there are probably more diseases that this could be considered potentially as important mechanistically, and certainly I think that that'll be an area of ongoing exploration. So I'm just going to wrap up there. I'll make a conclusion. First of all, we don't know everything about what neuropilin's doing during normal human biology. We certainly don't know everything about what it's doing during sarcoidosis. But so far we know that it's expressed in granulomas, that it's upregulated as monocytes differentiate into inflammatory macrophages, and that when you antagonize or that when you bind neuropilin with ligand, efzofitimod, that you're able to downregulate several cytokines that we think are important in sarcoidosis, and in fact we saw downregulation of those in a dose-dependent manner during the clinical trial. We know that it has very high selectivity for neuropilin-2. I think that that's good when you think about safety and toxicity. And finally, I want to point out that the initial trial that was done is very small. It needs to be reproduced. But I do think that these are good enough data that it leads us to the idea of having a registrational trial in sarcoidosis. And so that's exactly what's happening. It's the first ever registrational trial in sarcoidosis. I also want to point out the importance of supporting science when we think about how we're going to do clinical trials in sarcoidosis. And I would suggest to you and ask you that each of us, when we think about prioritizing clinical trials, supporting clinical trials, enrolling patients in clinical trials, think very carefully about the science. And we should really ground our clinical trials on which have good scientific rationale, but we should also make sure that sponsors are pursuing science as they pursue their clinical trials. It's not enough to just repurpose a drug and take it off the shelf without doing science. Those two things have to go hand in hand. And I know that many of you in this room feel the same way as I do about that. So I think I'll stop there, and hopefully we have some time still for questions. If you're interested to ask anything, there's a microphone right there.
Yeah, thank you so much for the. I want to ask about, you mentioned pulmonary sarcoid. How about patients with extrapulmonary sarcoid? Any of the patients there? And you think is it the same disease we have in such patients?
Yeah, so the question is about extrapulmonary sarcoidosis. And I think the first thing about it is that the trial is designed and measured for pulmonary sarcoidosis, and that was the focus of this trial. I don't think that there's necessarily any reason to think that this approach would not be effective in extrapulmonary sarcoidosis, but I do think that there are definitely biologic differences between pulmonary sarcoidosis and extrapulmonary sarcoidosis, and that we see responses that are different between agents in some of those things. For example, TNF antagonists work like a dream for skin disease and for neurodisease. They're much less effective for things like pulmonary disease, in my opinion. Some organs have more propensity to fibrosis. In the particular trial that I talked about earlier, we did look for development of new extrapulmonary disease. There was one person that got a flare-up of ocular disease, not clear whether it was sarcoidosis or not. But of course, during the registrational trial, we'll have to look at extrapulmonary disease, no question.
The placebo and the patients, were they matched regarding the stage of the sarcoid disease, the pulmonary?
Matched for scatting stage.
Stage 1, 2, 3, 4.
They all had to have parenchymal disease, so they were all stage 2 or 3. There were some differences, baseline differences in FVC, for example, as you can imagine with a small trial. But even if you do baseline adjusted analysis, you still saw the effects that I showed you. Oh, here's one.
Thank you, Daniel, for the talk. My question is about, because we are starting this project trying to reduce the dosage of corticosteroids, but my point is, we know that corticosteroids may have side effects. Why don't we start with, in the future, with just these new therapies? Because we are in the precision medicine era, no? And we are doing corticosteroids, immunosuppressive, but we are not personalizing the patients. So I know for legal reasons, well, we have to start at some point, but I think that maybe in the future we can think about it. No.
Yeah, I think that's a great point. And you bring up a couple things. One is, why do we start with steroids either in clinical treatment or in clinical trials? And maybe we should be enriching patients that are going to have progressive or bothersome disease and start right off with, and if we were better at doing that, I think we would. The second thing about precision medicine is I think we need better biology understanding. And then to really validate precision markers, you have to have a validatable endpoint. And I don't think we have that yet. Without a validated endpoint, it's very hard to validate biomarkers.
Hello, Daniel. Thank you for this wonderful presentation. My question is about concomitant sarcoidosis. Whether this drug can be given to concomitant sarcoidosis, both, that patient having both sarcoidosis and tuberculosis kind of situation?
Yeah, I guess, you have to wrestle with tuberculosis. If you're in India, you have to wrestle with tuberculosis a lot more than I do. And there are certainly biologic differences on a gene expression level and, of course, histologically between tuberculosis and sarcoidosis. So I don't know, and I'm not sure I could speculate about what would happen if somebody with tuberculosis got this medication. Of course, we'll have to monitor infections in general on the medication and see, what happens. With TNFs, I think it took until we got into clinical practice to understand that TB was going to be a problem. I don't know that when you have autoimmunity or genetic defects in this particular area of biology that we see a lot of tuberculosis, we know in TNF knockouts that they're completely unable to control mycobacterial disease. So, I think maybe more preclinical work will have to go into that.
In our population, we see more of like patient having gene expert positive, but non-necrotizing granuloma with all the features are showing sarcoidosis, but having gene expert positive, we consider them as a concomitant sarcoidosis and tuberculosis. In that case, we generally give ATT and corticosteroid. But my question whether with ATT, this drug, does it react or cross-react or some, what will happen with that?
I'm skeptical that this will be any worse for infection risk than steroids. If you really look at rheumatology databases, steroids in general have as many infections as any other immunosuppressant that we use. Of course, there are some special examples like rituximab causing certain infections, infliximab causing certain infections. But I think in general, steroids are just about as bad as it gets for causing infections.
Dan, thanks. I have two questions. And if you or maybe any of the HR scientists here has some information about this, I think it'd be interesting to the group and to me. The first is, do we know if steroids modulate expression of neuropilin-2? And I'll ask the second question after we hear an answer to that.
I'm going to punt on the first question, Peter. I don't know if we have flow on patients at different levels of steroid dose to know. Anybody know that? We'll get back to you.
Okay. And the second question is also probably one that HR scientists may have some information about. But for other diseases in which monocyte-derived macrophage recruitment is essential to the pathophysiology, and there are many, but rheumatoid arthritis, for example, would be one. Do we know if this pathway is upregulated in the macrophages, in particular in, say, rheumatoid arthritis or others?
We know some things. We'd like to kind of get that information out in the future. So maybe you have to join us in Milan for some of that information.
One up here.
Is neuropilin expressed in other granulomas like TB and hypersensitivity pneumonitis and things like that?
I don't think you stained. Did you stain for that, Sanjay? I'm not aware of that.
We've only really looked at the sarcoid granuloma at this point. There's some anecdotal evidence in sclerodermatous plaques, but we are following up on that. Neuropilin is seen in alveolar macrophages in, for example, neutrophilic asthma patients. Also in the hot joints of RA patients, sometimes neuropilin is seen during an active flare. But these are things we're looking at more closely. We haven't looked.
Okay, I think we're at the time. I want to thank everybody, and come on up if you have any more questions.